This doesn't look like it's working. Oh, it is working. Great. Starting to worry if this is going to work. Sarah or Alina, can you hear me? Hello? Sarah or Alina, could somebody confirm you can hear me? I want to make sure this is working because I got into it in an unusual way. My Zoom meetings were actually empty today in Canvas, so um, I want to confirm you guys got in the normal way. Can somebody please answer me? Sarah or Alina? You can type if you wish. Your mic doesn't work. Uh-huh. All right. But you are hearing me. Is that correct, Lena? And did you get in the class in the normal way? All right, let's go then. Um, thank you. Let's begin the class. So let me share my screen. I was worried that people weren't getting in the normal way because I didn't get in the normal way. And for some reason, all my Zoom meetings are blank. I can actually show you that right now. Yeah, no, I can't. Gotta go to Zoom.
Professor,
Uh-huh.
so I just tried joining this meeting by the Zoom meeting page on Canvas, and it's like nothing's coming up. It says no data.
That's what I got too. There's nothing there in the Zoom link. And so what I did was I actually called up an old class, and I don't know if this is going to be the wrong date, but I'll change it on the in the thing. Anyways, you got into the class. That's good. Like I said, are you seeing my screen here? Yeah, you are.
Yeah, that's
So
exactly what
that's
mine
what
looks
I'm
like.
seeing. And so I couldn't get into the meeting in the normal way, and I don't know what's going on. Um, this has been working previously for a couple of terms. I knew they turned it off once, but I thought they turned it back on, and I haven't gotten an email saying they turned it off. So I don't know. Anyways, we're in class. Let's begin. Let's state that today is February 19th. We're supposed to be on chapter 8. In fact, we're supposed to be well on chapter 8. We are actually on the last slide of chapter 6. So let me talk about that, and then we'll start chapter 8. So we were estimating the bacterial numbers by indirect methods. And there are two other methods I need to talk about: metabolic activity and the dry weight method. In metabolic activity, you're measuring some metabolic activity of the cells, such as the acid they produce or the CO2, and you're assuming that the bacterial metabolic product accumulates in direct proportion to the number of bacteria present. I've done this where you're measuring the amount of CO2 accumulating. And then what you do is you uh, make a graph of it, and then you can use this graph based on the amount of CO2 the bacteria produce for estimating the number of cells. And then the dry method um, is used when methods are not satisfactory, like filamentous bacteria and fungi. Organisms are filtered from the growth medium, and then they're dried and they're weighed. And then you look on a chart to determine how much weight is how many cells. So you get, I don't know, 1 gram, it'll tell you how many cells you have. Any question about any of that? The advantage of the dry weight method is you can dry down the cells and then you can weigh them whenever you have time. It's not um, an immediate thing. Any questions about estimating bacterial numbers, either by indirect methods or by direct methods? If not, let's close down chapter 6 and begin chapter 8. Here I'm giving you the major goals and a rough outline of chapter 8, genetics. Know the terms of this chapter. That's the first thing. 2, be able to describe what DNA replication is, RNA transcription, and protein translation. And then 3, understand the different types of mutation. We'll talk about all of these. Initially, we're going to talk about the structure and function of the genetic material, talking about the flow of genetic information. We'll then talk about DNA replication, move on to RNA transcription, and then lastly cover protein translation. We'll then switch gears and talk about the regulation of bacterial gene expression using operons to talk about how gene expression occurs. We'll then move on to the topic of mutation. What is mutation? How it occurs? We'll talk about genetic transfer and combination. And lastly, we'll talk about genes and evolution. Any question about any of this? All right. Genetics is the study of what genes are, how they carry information, how that information is expressed, and how genes are replicated. A gene is just a segment of DNA that encodes a functional product. It's usually a protein, but the gene could also code RNA instead of protein. So here you're looking at one gene on a chromosome, and it's that part of this chromosome. And then you'll notice in a eukaryote chromosome, we have regions that are not genetic material, non-coding DNA. And then we have a gene too. Now in bacteria, most of the genetic information codes for a gene. There's very little non-coding DNA. But in eukaryote DNA, most of the DNA is actually non-coding DNA. Here we're looking at a chromosome and it's showing you, let me blow it up a little bit, showing you the different genes that have been mapped to this chromosome, their location, what the genes are, and you can see their different locations on the chromosome. Any question about any of that? Oops, why is that not working? Not sure what happened there. Let's move that up here. Terminology. Genome is all the genetic material in a cell. When we're talking about an organism, I mean a microbe, it would be all the genetic material in the organism as well. The study of genomes is called genomics. The genotype are the genes of an organism. The phenotype is the expression of these genes. For example, an individual who has blue eyes has two lowercase b genes because the lowercase b allele codes for blue eyes. So the phenotype is blue eyes if you have the genotype lowercase b. However, individuals who have the phenotype brown eyes, shown here, can have the genotype capital B or and lowercase b, or they can have the genotype capital B, meaning they can have both genes capital B, or they can have one capital B gene and one lowercase b. You'll note that the lowercase b is recessive and the uppercase B for brown eyes dominates over the blue eyes. Any question about any of that? I don't know why this is not working when I click on it initially. Here we're looking at a chromosome from E. coli, and the DNA is on the chromosome. Obviously, there's proteins in it as well. You can't see the DNA from the protein, at least in a light microscope or even this electron microscope. But the Gene would just be one segment of this DNA. Any question about any of that? All right, DNA is a polymer of nucleotides. They are adenine, thymine, cytosine, and guanine for DNA, or you could call it A, T, C, and G. I don't care which name you know, but you got to know one. Okay? DNA is a double helix with the associated proteins making the chromosome. So the DNA and the proteins make the chromosome. DNA is a double helix. What we mean is it has two strands of DNA, one here and one there, and they are linked together only by hydrogen bonds between each strand. And they're actually the hydrogen bonds only occur in the uh, nit- nitrogenous bases. What is holding the two strands of DNA together is only hydrogen bonds, but one strand of DNA is held together by the deoxyribose phosphate sugar backbone, meaning here we have a sugar, it's connected by a chemical covalent bond to this phosphate, which is connected by the chemical covalent bond to the phosphate, and then this sugar phosphate backbone holds the single strand of DNA together. That's a very stable link holding one strand together. The link holding the two strands together is very tenuous. In fact, the hydrogen bonds break and reform all the time at our body temperature, meaning 37 degrees Celsius. The hydrogen bonds form only between A and T, and there's two of them between A and T, or between C and G, and there's three hydrogen bonds between C and G. Any questions about any of that? All right,
So,
lastly, go on.
so, so the nucleotide is the bases, the thymine, cytosine, and uh, or what you're calling A and T and C and G.
Yes, the nucleotides are A, T, C, and G, and these are only these right here, the nitrogenous bases. They have nitrogen in them, they're called nitrogenous bases, by the way. Let me add that the two strands of DNA do not run in the same direction. They are anti-parallel, meaning they don't run this way. They run that way, where one has the 3' end on this end and the 5' end on this end. And here we have the 3' end here and then the 5' end here. The 3' and the 5' are just biochemists' way of talking about the carbon. And this carbon here is the 3' carbon, and that carbon there is the 5' carbon, meaning the fifth carbon in this sugar. Any question about any of that? You don't need to know the 3' and the 5', but you should know that the two strands of DNA are anti-parallel, meaning they run in opposite directions. All right, let's talk about the central dogma of biology. Oops, lost my place. There we go. And that is uh, the information in DNA starts obviously in DNA. DNA codes for the information. That information is transcribed into RNA. And then that information is translated into the information of the protein. So the information flows from DNA to RNA to protein. That is known as the central dogma in biology. Each of the information is stored in their own language. The DNA information is stored in the DNA nucleotides. the information in RNA is stored in the RNA nucleotides, and the information stored in the protein is stored in the amino acids. So there's different languages storing each information. Why is that there? Let's talk about the flow of information, genetic information. You can have the flow of information flow within a cell, between generations of cells, and between cells of the same generation. Within a cell, DNA flows, the information in DNA flows to messenger RNA, which then flows to protein. So this is the flow of information within a cell. You can have the flow of information happen between generations of cell, where the DNA in one parent cell flows to the daughter cells, meaning the DNA replicates, divides in two, splits down the middle, and then one daughter cell gets one copy of the DNA, the other daughter cell gets another copy of the daughter DNA. Now, for humans, we have two parents who get together, and then the DNA flows from the parents generally to one offspring. And that happens by sexual reproduction. So a little different, but similar enough, meaning the both parent will separate their DNA and make their gamete cells, and the gamete cells recombine to make the offspring. Any question about any of that? We can also have the flow of information flow from one cell to another cell, and that's called between cells of the same generation. Usually the DNA leaves one cell and then can be actually naked DNA out in the environment, which can be taken up by a recombinant cell. This recombinant cell can then express the genes on that DNA, getting genes from this cell. So the cell gains DNA from another cell. When this happens, if the DNA leaves this cell, the cell may die. And that's why the DNA leaves the cell. It doesn't always happen that way, but it may. And we'll talk about that when we're talking about the flow of information between cells of the same generation. Any question about any of these? All right. In the vertical flow of information, we've stated that the information flows from DNA to RNA to protein, and that the language is 4 DNA nucleotides, meaning there's only 4 letters in the alphabet of DNA. And actually in RNA, there's also only 4 letters in the alphabet, but this is a little different language because this is RNA nucleotides. And then there's 20 different amino acids. And so there are 20 different letters in the alphabet of a protein, 20 different amino acids, at least for humans. And actually most organisms also have 20 or very close to it, 20 amino acids. Some organisms which have more or a different amino acid, they tend to drop an amino acid so that most organisms keep about 20 amino acids for the letters in the alphabet of proteins, meaning they only keep 20 amino acids in their proteins. In comparison, English has 26 letters in its alphabet. Another way to say it is when the information is stored in DNA, you could say the information is stored as, that is a dog. When the information is stored in RNA, it's stored in a little different dialect, and you'd say something like, that is a dog. And when the information is stored in protein, it would be something like, quello è un cane, a totally different language. Any question about any of that? All right, we talked about the flow of information within a cell. Let's talk a little bit more about the flow of information between generations of cells. This is known as the vertical flow of information. DNA replication is semi-conservative. That means one strand is used as a template to make a new strand of DNA. And how that happens is the DNA polymerase binds to the template, let's go back here, sees a T here, and then puts on an A there, because it only puts on the complementary base, and A is complementary to T. And then the DNA polymerase moves down to the next nucleotide on the DNA, sees a G here, so it adds a C. The C complements the G. And then it moves down the template to the next base, sees a C there, and so it adds a G because G complements a C. And it just keeps moving down. Here we have an A, so we'll add a T. And that's what we mean by DNA is semi-conservative, because when the DNA divides this way, one strand of DNA is the original DNA serving as a template to make the new strand of DNA. And then the other original strand of DNA serves as a template to make the new strand of DNA. And then the cell will divide with this strand, double strand, becoming the genome of the next cell, and this double strand becoming the genome of the next strand. That's what semi-conservative means. That is different from conservative replication. Conservative replication is that one cell would get both strands of the original DNA, or the dark strands, and then the other strand would get both new strands of DNA or both light strands of DNA. That's the conservative model of DNA replication, which does not happen. The semi-conservative model is that one strand of DNA, the original, goes to each daughter cell. And so each daughter cell has one original strand of DNA and then one new strand of DNA. That's the semi-conservative model. Don't ask me what the liberal model is. Any question about any of that? And how that happens, the semi-conservative model is the DNA polymerase just uses one strand as a template, sees a base here, and puts on the correct complementary base, the one that binds to A, which in this case would be T. What drives the reaction is that this T being added is a triphosphate, and then two phosphates come off. That releases a lot of energy, which then gives the reaction the energy it needs for this phosphate to bind to that sugar, deoxyribose. Any question about any of that? So DNA has more energy than the smaller molecule DNA, and then in this case, the T triphosphate, and that energy is obtained from breaking off these two phosphates. Both of these are high phosphate bonds. You break the bond here, and this T then has the energy it needs to bind to this sugar. Let's talk about DNA replication by watching a video of DNA replication. No commercial.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about this video? Oh, what the heck is going on here? Oh, I see. All right, so let's talk about DNA replication. Let's end the show here. Keep losing my place. DNA replication happens differently on the two strands of DNA, and that's because the DNA polymerase can only move in the 5' to 3' direction. And so on the leading strand, the DNA polymerase is moving in the correct direction. And so in theory, the DNA polymerase could begin at the start of the chromosome and then move all the way to the end of the chromosome, adding the correct nucleotide base corresponding to the template DNA. Any question about any of that? Now, in theory, that could happen, but most of the time, the DNA polymerase begins, and then for some reason it just falls off on its own, and then another DNA polymerase will bind here, and then continue the process. Let me state the process begins because of helicase. The little video mentioned there are There are some enzymes which are very important. Helicase is one of the 3 enzymes you do need to know about. Helicase opens up the 2 strands of DNA to make it single strand DNA. You need single strand DNA to replicate the DNA. So you need helicase to open up the double strand DNA to make it single case. The other enzyme, or the second enzyme that's important, is the DNA polymerase. It will bind to the single strand DNA, using it as a template to make the DNA double strand, meaning it'll put in the correct nucleotide. And on the leading strand, this DNA polymerase is moving in the correct direction. And like I said, in theory, if the DNA polymerase never falls off, you may only need 1 DNA polymerase and then transcribe the entire chromosome. Now, in reality, the DNA polymerase often falls off, and so you can have more than 1 DNA polymerase making the new strand of DNA. Any question about any of that? On the lagging strand, the DNA is not in the correct direction. It is opening from the helicase this way, but DNAs cannot be replicated this way. It can only be replicated this way, meaning in the 5 to 3' direction. And that means that the DNA polymerase is running in the wrong direction, and it makes the new strand of DNA in short segments called Okazaki fragments, after Dr. Okazaki, who discovered it. And what happens is, for this batch of DNA, an Okazaki fragment, DNA polymerase binds here, and then moves 5 to 3' direction this way. And then one DNA polymerase, another one, binds here and makes an Okazaki fragment, making the double strand DNA this way. And then you have this repeated all the way on this single strand of DNA. So there's many different Okazaki fragments because they're in short chunks. Whenever the DNA is made by another polymerase, you have a gap in the DNA. Like, let me see if I can blow this up. Come on. Not working for some reason. Oh, there we go. So here, when this comes up to here, it won't bind this newly synthesized DNA to this new strand of DNA right here, meaning this Okazaki fragment right there. That won't bind. And there, they're actually showing it to you. It's not linked right there. And it's not going to link right here and there either. So whenever the DNA comes on and makes a strand of DNA, there will be a gap in this single strand base of DNA where this DNA isn't linked to that DNA there. And what will connect that together is ligase. So ligase will bind to this. Let's talk about it right here. Ligane will bind to this Okazaki fragment and that Okazaki fragment, and then make that gap filled in, not with the nucleotide. It'll just make the phosphate of this DNA nucleotide bind to the ribose of this DNA nucleotide. Any question about any of that? So ligase is working all the time on the lagging strand of DNA. For each Okazaki fragment, you're going to need ligase to bind twice, although that one binding to that one, that'll be the same, although this one will still need 2. You'll need a ligase here and a ligase there. On the leading strand, if the DNA polymerase never falls off, the ligase does not need to work at all. However, if the ligase, the DNA polymerase falls off, and then another one starts here, ligase will need to connect this strand of DNA to this strand of DNA, which will be synthesized here. Is that clear to everyone? So ligase does work occasionally on the leading strand, and then it works on the lagging strand all the time. I think that's all you need to know. Ligase, DNA polymerase, and helicase. You don't really need to know about primase, putting on the primer. Whenever you start a DNA segment, you have to start it with an RNA primer, but that RNA primer will be removed, and then that'll be filled in with DNA. Any questions about how DNA replication occurs? All right, I've stated that DNA is made only in the direction 5' to 3' direction. It's started by an RNA primer. The leading strand is synthesized continuously. And in theory, a DNA polymerase could replicate the entire chromosome for the entire single strand of DNA. But in reality, that DNA polymerase will tend to fall off. The lagging strand is synthesized discontinuously, resulting in Okazaki fragments. And you need the DNA ligase to connect the breaks in the new strand of DNA on each Okazaki fragment, or wherever the DNA polymerase falls off on the leading strand. All right, any questions about any of that? If not, let's move on to RNA transcription. Transcription is the synthesis of RNA from DNA. Unlike DNA replication, where the entire genome is replicated, in transcription, only the genes are transcribed. Any questions about any of that? So in RNA transcription, you do not transcribe the entire genome. You only transcribe the genes. And remember, in eukaryotes, most of the DNA does not code for a gene. I shouldn't say most, but the majority of the DNA does not code for a gene. DNA is transcribed to make RNA, and we have 3 different types of RNA that you need to know about. There's actually more, but you only need to know about 3 types of RNA. There's messenger RNA, or mRNA. There's transfer RNA, or tRNA. And then there's rRNA, or ribosomal RNA. Each of these 3 types of RNA is important in translation, protein translation. And we'll talk about what they do when we talk about protein translation. Transcription begins when RNA polymerase binds to the promoter. The promoter is a region of a gene upstream of the gene, and it codes for the RNA polymerase to bind here. So that's what the promoter does. And then the RNA polymerase moves down the DNA, transcribing it. Transcription, once again, proceeds in the direction only 5' to 3'. So RNA is made in the direction 5' to 3'. And the RNA polymerase continues transcribing the DNA until the RNA polymerase reaches the terminator. The terminator is a region of a gene downstream of the gene, and the terminator codes for the RNA polymerase to fall off the DNA. Once the RNA polymerase falls off the DNA, that will release the RNA, and then it can, well, in a eukaryote, it can then move out of the nucleus and go into the cytoplasm and a prokaryote that's already in the cytoplasm. Any question about any of that? Well, here's a question for you. If the RNA polymerase is transcribing the DNA, how does the RNA polymerase know whether to make messenger RNA, tRNA, or rRNA? Can anyone answer that? That might be a question on a quiz. Nobody can answer it?
Is it,
Go on.
I was gonna say, is it based on the location of where it's at or like which step it's in?
You talking about which gene it's at in the chromosome, or are you talking about something else?
Something else, more like what part of the actual transcription it's on?
You're close, but not correct. Actually, if you had said what gene it's on, that would be the correct answer. And
Oh,
that is
okay.
the RNA polymerase does not need to know what RNA to make because the gene itself will code either for messenger RNA, tRNA, or rRNA. There's only a handful or less genes that code for tRNA, and also a handful or less, or maybe 24 or less. Let me think about that. I do know this. It's actually a handful of genes that code for rRNA, and then about a dozen or less. Is that right? No, actually, it's about 20 or less genes that code for tRNA. And then there are most of the genes code for messenger RNA. And it just depends on what gene it is, what it'll code for. If it's a tRNA gene, then the RNA polymerase will make tRNA. If it's an rRNA gene, then the rRNA polymerase, the RNA polymerase will make rRNA. And if the gene codes for a protein, the RNA polymerase will code for messenger RNA. Any question about any of that? So the RNA polymerase doesn't need to know which RNA to make because the DNA itself codes for which RNA to make. And the RNAs are all the same. They're the same 4 RNA nucleotides. The only difference is the sequence of RNA nucleotides. If the sequence codes for a tRNA, the gene will be transcribed as a tRNA. If the gene codes for rRNA, then the gene will be transcribed as rRNA. But if the gene codes for a protein, the gene will be transcribed as messenger RNA. Any question about any of that? All right. Here we're actually looking at, well, it is electron microscopic image, but this is actually a fluorescence microscopic image as well. And here we're looking at RNA polymerase. That's actually DNA there. And there is the beginning of RNA being transcribed. And here we're looking at it here, DNA being transcribed into RNA, in this case, messenger RNA, which will then later be translated into protein. Obviously, only messenger RNA will be translated into protein. Any question about any of that? Let's go on to the next slide. And we have a little video to watch on RNA transcription. So let's watch it. Assuming I can get it to work. Nope, didn't work.
Bundle and save on insurance. Libbity
After the
bibbity.
commercial.
Swoon.
Swoons? Oh, I love those birds.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein.
I'll talk about that in a little bit. Let's not talk about it yet. All right, any questions about any of this? Let me show you a picture back here. Here we go, right here. You'll note that initially when the RNA is being transcribed, it's actually hydrogen bonding to the DNA. However, oops, go back a little. That hydrogen bonding only is at the start of RNA transcription, meaning the newly transcribed RNA hydrogen bonds to the DNA. But the later nucleotides of RNA actually fall off the DNA. That is important because that means this DNA is single-stranded, and that will allow this single-strand DNA to bind to the single-strand DNA up here, and then the DNA becomes double-stranded after that. And so in RNA transcription, you have the helicase making the DNA single-stranded, but the only region which is really single-stranded is the region before the RNA polymerase and after the helicase, and then a little region after the RNA falls off the DNA. And then this becomes double-stranded again. Any question about any of that? All right, so RNA transcription begins when the RNA polymerase binds to the promoter. Remember, the promoter is the region of the gene upstream of the gene proper. And then the RNA polymerase moves down the DNA, transcribing the RNA. Initially, as shown here, let me blow this up. As shown here, the RNA hydrogen bonds to the DNA. But then the RNA falls off, and so we have a little region of single-strand DNA. That single-strand DNA can then hydrogen bond with the other strand of single-strand DNA, forming double-strand DNA. Any question about any of that? And then the RNA polymerase continues moving down the DNA, transcribing the RNA. And as it moves, the two strands of DNA rebond to each other. So there's only a little region which is single-stranded. And the RNA polymerase will continue doing that until it reaches the terminator. The terminator is a region of DNA after the gene proper. So you can say it's part of the gene, but we're going to say it's after the gene proper. And the terminator codes for the RNA polymerase to fall off the DNA. that will release the RNA strand and as well as release the DNA strand. And then when that happens, the two strands of DNA can snap back together, hydrogen bond, and become double-stranded again. Any question about any of that? And there I've just shown where the terminator has the RNA polymerase fall off, the RNA transcript falls off, and then the DNA snaps together. The two strands snap together. All right, any questions about RNA transcription? If not, let's move on to protein translation. Translation is the synthesis of proteins by ribosomes using the information in the messenger RNA. Messenger RNA is translated into codons, and so you need a group of 3 nucleotides to code for 1 amino acid. And that's why we say 3 nucleotides is a codon, because the messenger RNA is read in groups of 3 nucleotides that we call a codon. Any question about any of that? And here we're looking at the flow of information leaving DNA, being transcribed into messenger RNA, which will then flow into the information of protein during protein translation. Although the messenger RNA codes for the protein, translation does not begin at the start of the messenger RNA. The ribosome has to move down the messenger RNA, and it does not begin protein translation until the ribosome reaches the start codon, which is AUG. So you should know this, that the ribosome moves down the messenger RNA, and it doesn't really begin protein translation until the ribosome reaches the start codon, which is AUG. That is the start codon. You must know this. You should also know the start codon codes for 2 things. One, it tells the ribosome, begin protein translation here. And 2, it codes for the amino acid methionine. So AUG always codes for the amino acid methionine. And that means all proteins that get translated have to start with the amino acid methionine. What do you think a cell does if it makes a protein, but it doesn't want the protein to begin with the amino acid methionine? What do you think the cell does?
Does it excrete it?
It just removes it. That actually happens in the Golgi apparatus. So if the protein doesn't want to begin with methionine, that amino acid has to be removed from the protein. Because all proteins start with methionine, because that's the first start codon. And then the ribosome moves down the messenger RNA, reading it in groups of 3 called the codon. And for each codon, it will add another amino acid. And this will happen until the ribosome reaches 1 of the stop codons. You should know there are 3 stop codons. I'm not going to require you to remember what the nucleotide sequence is for a start codon, but there are 3 of them. And when the ribosome reaches a stop codon, protein translation stops. The ribosome actually falls off the messenger RNA. If there's a tRNA around, it'll be released. And then the protein is released. And in a prokaryote cell, it could be actually become useful. In a eukaryote cell, Well, this is happening in the cytoplasm, so I guess it could happen right away either. Any questions about stop codons, or start codons?
So what reads the mRNA and brings the correct amino acid to the growing protein is the ribosome?
Not exactly. People say the ribosome reads the messenger RNA. That's not true. Strictly speaking, I'll talk about that in the next slide. It's the tRNA which is reading the messenger RNA, but the ribosome does move down the messenger RNA.
Okay, so then what were the two things that the AUG codon codes for? It was something about a ribosome and it
No,
codes for...
it tells the ribosome begin protein translation here at the start codon, and then it also adds the amino acid methionine. So AUG serves two functions. It does code for the amino acid methionine, but if it's seen the first time on the by the ribosome, it will begin protein translation. Okay, so
Okay.
the first time the ribosome reads AUG, it will begin protein translation. But if protein translation is started and the ribosome reaches an AUG again, it only encodes the amino acid methionine. Any question about that? All right. All right, let's talk about the genetic code. I mentioned that the messenger RNA is read in groups of three called the codon. And each codon codes for one amino acid. But organisms contain 20 or about 20 different amino acids, but there are 64 possible codons. Why? Why are there more codons than amino acids? And how does the cell handle that? Well, the cell handles that by having more than one codon code for an amino acid. For example, phenylalanine has two codons coding for one amino acid. Both UUU and UUC code for the amino acid phenylalanine. Serine, on the other hand, has four codons coding for that same one amino acid. Leucine actually has six codons coding for that same one amino acid. So most of the codons have more than one codon coding for one amino acid. I guess I should word that. Most of the amino acids have more than one codon coding for it. But there are two exceptions, and that is methionine is only coded by AUG. And then the other one you don't need to know about, the amino acid tryptophan is only coded by UGG. All the other amino acids have more than one codon coding for that amino acid. Any question about any of that? This difference between 20 amino acids but 64 codons is known as the degeneracy of the code. It's an older word of degenerate, just meaning that there's more than one thing coding for the amino acid. Any question about any of that? All right, let's talk about translation video. Let's go watch a little video on that. Or turn it down at the start.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Any questions about that video? Want to be right about here, right about there someplace. I'll come back to this. Let's see if I can shrink this. There. No, that didn't work. Oh, here it is. So there are 3 parts needed for, well, actually 3 RNAs needed for protein translation. You need the messenger RNA because it has the code for what we're going to make. And usually messenger RNA codes for, actually messenger RNA always codes for a protein. And so it has the codons that code for the amino acid of the protein chain. You also need tRNA, and it brings in the correct amino acid. How it does that is tRNA has an anticodon, which actually reads the codon of messenger RNA. And that's why it's really tRNA, which is reading the codon, not the ribosome. And tRNA brings in the correct amino acid, which means that this codon codes for that amino acid. And how it works is that this anticodon links with that amino acid. And then the third RNA, which is required in protein translation, is ribosomal RNA, which together with ribosomal protein makes the ribosome. Any question about any of that? So remember, the ribosome is made up of rRNA, or ribosomal RNA, and ribosomal protein. All right, let's begin. So the ribosome has two sites on it. You don't need to know the name of them, but the first site is where the first tRNA binds, and the ribosome actually stabilizes this interaction. The hydrogen bonding between U, A, and C would be very weak to A, U, and G, meaning there is only what, 2, 4, and then 7 possible hydrogen bonds. Let's see, 2, 4, yeah, 7 possible hydrogen bonds. That would not be enough to link the tRNA to the messenger RNA. But the ribosome right here in this area here has a core that the tRNA comes in, and the ribosome stabilizes this interaction. The ribosome also moves down the messenger RNA, and that's why the tRNA can bind to this, and that's because the ribosome moves down the messenger RNA, and now this first channel, let's call it that, in the protein is now open to the first codon, which is always AUG, because AUG codes for the starting of protein translation. The ribosome has a second channel in it, and it allows the next tRNA to come in. And the only tRNA that can come in is one that has the anticodon AAU, because the second codon has the UUA codon. And obviously this will vary on what amino acid we're coding for, depending on which protein we're making. But for this one, it's going to be UUA. And the ribosome, once again, stabilizes this tRNA interaction so that this tRNA can come and bind to the second codon and then bring in the correct amino acid. Any question about that part? Now, the next thing that happens is the amino acid in the first channel of the ribosome, or the first hole in the ribosome, what do you want to call that, is removed from the tRNA. And then that amino acid is added to the amino acid in the second channel of the ribosome. And that's going to begin the growing polypeptide chain, which will eventually become the growing protein, which will eventually become the finished protein product. How this is released, this amino acid, and then bound to this amino acid here, is by an enzyme on the ribosome, and it's actually an RNA enzyme or a ribozyme. And this is the only case of a RNA enzyme that we will talk about in this class. The only time we'll ever talk about an RNA enzyme. Well, there might be another time, but generally the only one I'll hold you responsible for. And that is this enzyme working that is actually an RNA enzyme, and it's part of the rRNA of the ribosome. Any question about any of that? And Tutora doesn't mention it, by the way, that this enzyme here is actually an RNA enzyme. Now, when the amino acid is removed from the tRNA, that tRNA can then fall out of the ribosome, and that is then a signal for the ribosome to move down one codon. So the first amino acid moves out, and the first tRNA is released. That allows the ribosome to move down the RNA by one codon. And you'll note that Now what used to be in the second position of the ribosome has moved into the first position of the ribosome. So that must have a connection there. And then this is in the first position. That allows the second position to take on a tRNA, and it will be the tRNA that contains the amino acid for GGU, because that's what this codon is. And that records the correct amino acid. So once again, the tRNA is reading the code of the messenger RNA and then bringing in the correct amino acid. When that happens, the ribosomal enzyme, the rRNA enzyme, removes the amino acids from this position in the ribosome and then attaches them to the newly added amino acid in this position of the ribosome. Any question about any of that? That will then allow this tRNA to fall out, which will be a signal for the ribosome to move down one further codon. And you can see we have several amino acids added, and the amino acids have been removed from this tRNA and then added to the newly added amino acid, which in this case was actually the amino acid methionine. So here methionine is only coding for, AUG is only coding for the amino acid methionine. It's not coding for start protein translation here. But in the first one right there, it coded for both. And there's the first amino acid there. Anyways, the ribosome continues moving down the messenger RNA, freeing up the second codon, moving from the second codon to the first, I shouldn't say codon, the first position of the ribosome, moving from the second position of the ribosome to the first position of the ribosome. And that process continues until the ribosome reaches a stop codon. Stop codon tells the ribosome stop protein translation here. That will make the ribosome fall off of the messenger RNA. That will release the amino acid chain, releasing the protein. And then that messenger RNA is then free to begin the process of protein translation again, where a ribosome will begin at this start of the messenger RNA, and then read down it to once again translated protein. Any question about any of that? All right, they're showing you the ribosome disassociating from the messenger RNA, releasing the newly made protein, and also releasing the messenger RNA. Any questions about protein translation? Because that's the simple version. Let's talk about some complexities now. In both a prokaryote and a eukaryote, here we're looking at protein translation. Actually, we're looking at it here in a prokaryote cell. And as you can see, we have a longer well, I guess you can't say that's protein translation. Here we're looking at the transcription, and there we have a longer messenger RNA than this one, than that one. That one you can't see, but it's longer than that one. And that one, that one, and that one. And as you notice, the further you come this way, the smaller the RNA transcript is. And that's because RNA transcription is occurring. And here, RNA transcription is either finished or about to finish. And so we have a very long RNA transcript. Here, RNA transcription is beginning. And so we have a very small RNA transcript. And you do see this in eukaryotes as well as prokaryotes, that in one gene, you can have more than one messenger RNA made from the same gene. In fact, in this one, 1, 2, 3, 4, 5, 6, 7, we have 8 different messenger RNAs being made from the same gene at the same time. Obviously, that one started earlier, but it's going on at the same time as this one here. Any question about that? And then in prokaryotes, it's even more complex, and that is that before transcription finishes, you can have protein translation beginning. And here you see that this blue line is the RNA transcript being made, and then these red things on it are ribosomes, and this ribosome has begun, and so has this one begun, before the messenger RNA transcript is even finished. You have this happening in a prokaryote cell because all of this is happening in the cytoplasm. And so a messenger RNA is being made in the cytoplasm, and then the ribosome can bind to that messenger RNA, even when it's quite small, and begin protein translation. In fact, you're seeing here, this one has 2 ribosomes bound to it. That one has numerous ribosomes bound to it. Here we see 1, 2, 3, 4, 5, which is closer to that one there. So this one, I don't know what it is, 20 to 30 ribosomes bound to it, making 20 to 30 different proteins from this one protein messenger RNA. And then this gene has multiple RNAs being made at the same time, and obviously multiple proteins being made at the same time. You got 20 to 30 being made on this RNA, messenger RNA at the same time. And then you have all of these being made at the same time too. So that's a complication in prokaryotes where you have multiple messenger RNAs being made at the same gene at the same time, and you have multiple proteins being made at the same time, and it's more than just one protein being made on each messenger RNA. On this one, we have 1, 2, 3, 4, 5 proteins being made all at the same time. And that's just a complication you have in prokaryotes. Eukaryotes does have the complication of more than one messenger RNA being made on the same gene at the same time. They do have that, but they don't have this one. And that's because transcription occurs in the nucleus, translation occurs in the cytoplasm in a eukaryote cell. All right, any question about any of that? If not, I'm going to end it here, and I'll see you at 6:31 for the lab. Oops, let me go back and mark that. All right, I'll see you at 6:31. Bye.